to share the dais which has been shared by so many dignitaries and the scientific legends of our country. And is it okay? Okay. So I really thank the President of Bangalore Science Forum and the other members for giving me this unique opportunity. And also, as you just got to know from the President that I am interested to come out from the lab and contribute to the direction of the school children, those who are our treasure. Especially, I believe the treasures are stored in the village, original innovative mind, which are not contaminated. We have to get them in the forefront, and Bangalore Science Forum works for it. And I promise that I will be always with you for that work.
Thank you, sir.
So, it so happened that today I thought of when he called me, then I said, okay, I'm going to give a talk on the genes and disease. And when I was sorting out the genes and disease, the first thing comes in our mind is the most devastating disease is the cancer. Of course, there is a different other genetic disease, there are different neural diseases, and I happen to be getting to that pretty serendipitously. So I thought of one of the lectures which I often use in my this science workshop for the children. I thought I will make it little more and then I'll present in front of you. And that will be easier to convey three messages to you. The message one is how important this disease connected to our lifestyle and genetics. How important it is for us to understand the basic of disease for everyone present in this hall. And there are a lot of hope. There is no hope. Science is working for it. And we are progressing in this direction so that to talk about the most recent therapeutic advancement. So presenting the basic informations, I will try to present in a very lucid manner. I will try to, put you in a kind of a more easy manner. But any of you are expert of the field, you have to excuse me. And any of you not expert but would like to be expert, if I can help, my email, my mobile number, all are available in net. You are most welcome. And I promise that I will respond to you. So the first thing, if I have the lights off here, that will be very nice. So after I came back from Japan and United States, I established my lab in transcription and disease laboratory. In this lab, we try to understand how genes are organized, how genes function, and can this gene function be connected to disease. That was the philosophy. I tried to do that. In this context, we try to work on the oral cancer, which is the topmost killer of our country. We try to connect these observations with one of the important infectious disease, HIV, and of late you just heard from the President is that central nervous disorders. So, but today I'm going to talk to you about the genes and cancer. To begin with, if I have time, I'll show you a glimpse of the most recent discovery of my group, which we are working almost a decade on the neural disorders. So if you look at a very simple cell in the human body, when the cells is under full function and the resting, you will see each of our chromosomes. So human being, there are 46 chromosomes. Each of our chromosomes are present in a very three-dimensional definitive structures. If you open it up, then you will find that this white thing is the DNA, and this white thing, the yellow thing, is the protein core. So this DNA and protein core together forms a very organized structures. And this is the structure we call chromatin. And this protein and this DNA can harbor a very simple chemical modifications. You know this DNA sequence, what you find, that determines you is you, me is me. I tell you a very interesting story. You know, the Makkah for Science is posting Harvard Laboratory in the United States. So I was going for a meeting. And I got down at Swayasa station, picked up a cab to go to Coast Spring Harbour Lab. So this guy who was driving the car, he's a big ponytail, an American driver. So he is asking me, so what is this meeting about? I was hesitating. Should I tell this taxi driver what is the meeting about? I kept quiet. This guy is an offender. He said, is it about genes or bugs? I fall from sky, my God, what is he talking? He's asking me, is it about genes or bugs? Bugs means, you know, this Drosophila or anything. I could not, stop my curiosity. I asked him, do you know what is gene? So he turns his pony and he said, why not? Well, you is you and me is me. That's the gene. I never had a more simplified definition of gene anywhere in any textbook, any knowledge. Why you is you and me is me? That is controlled by gene. But more than gene, there is something else. What is that? That is not genetics, that is called epigenetics. What is epigenetics? As I just told you, this DNA and the protein, they get a very simple chemical modification. That modification finally determines your state of tolerance, your state of disease, your state of response to drug, and your state of mind. You know, one of the major causes of cancer, people say there are two major causes of cancer. One is age, another is irritation. Avoid irritation. And both the cases, your gene sequence does not change. What changes your protein and DNA modification? It is very important. I'll come to that later. That is why these modification, protein modification or DNA modification, is very, very important. That determines your lifestyle. You know, we are blessed because of the spices. The spicy Indians. Lot of flus, lot of disease, actually we avoid by taking the spice, by inducing our epigenetic changes. This is absolutely true, is proven today. Your cumin, your curcumin, your haldi, your rasam sambar powder, this has tremendous effects on you. Okay. So this is what the epigenetics is, basically the specific modification like methylation, acetylation, phosphorylation. Very simple chemistry happens on your DNA and protein. Very specific sequentially this happens, that determines your gene function. And you will be surprised to know that this knowledge of epigenetics and its connection to your disease, your different cancer actually alteration of different epigenetic marks. And that's why this has become a tremendous opener for the field for drug designing. We need new targets, and these all modification, what you see, there is a histone protein, there is a lysine 4, there is a histone protein, there is the arginine 17. This residue, protein residue, gets a specific modification that sets up a kind of, see, this is the histone H3 protein. I showed you that yellow core. There are 4 different histones there. One of them is, or 2 of them are H3 and H4. This has a specific targets of modifications. And this modification, it is kind of a telegraphic concept. Because you have said 10 amino acid straight, 2 gets methylated here, 1 gets acetylated here, 1 gets phosphorylated here. That establishes a kind of code. Okay? This code determines which gene will be expressed and which gene will not be expressed. This modification is actually done by a specific enzymes. In several cancers, for example in the oral cancer, one of the enzymes shoots up, indiscriminately modifies. That leads to a huge amount of upregulation of the gene. And you will be surprised to know that these enzymes get triggered up because of nicotine, which you put inside your mouth. Okay, so let's go to the term called cancer. So cancer, historical record, which Europeans and Americans believe comes from 300 BC. So Hippocrates named tumor is a carcinoma or carcinoma. So tumor basically spread the legs like a crab. That is what the symbol is used. But if you look at the much older literature which comes from India, okay, and this is the Sanskrit pronunciation of it. If you English translate it, basically in the Ayurvedic literature much before, thousands years before, there was a description which we call Arbuda. Arbuda basically in the later stage is actually the cancer description. And that is the oldest literature available in the world. Well, good to know that now the concept of Arbuda is being accepted all over. So normally people ask, what is cancer? The problem is cancer is not a single disease. It's 100 diseases together, you call it cancer. Not one disease. Okay? But they are a commonality. I will come to that commonality soon. But you must know it is the term used for the disease which abnormal cells divide. In our body, in our everyday life, cells are dividing all the time, depending upon the place we are talking about. You just now had a hot cup of coffee. If it is very hot, what do? You basically destroy your inside cells. It cannot remain in that way. It got immediately replaced. At your any age, from 1 year to 80 years, if you cut a finger, what happens? It immediately seal up. If it is a too big a cut, you get stitch and seal up. When it seal up, there is a cell division happen. Imagine the cell division start but don't stop. What will happen? You will have a terrible effect of this will start growing. So that means in our living system, everyday life, every day your blood cells are dividing. Your blood cell is getting destroyed. From your bone, the blood cells are made. So, but there is a regularity. The moment it is irregular, that is called cancer. You look at in the bone marrows. So this irregularity brings that. But there is a new concept of cancer. This concept I am confident that most of you haven't heard before. Cancer is called somatic cell pregnancy. You know, pregnant mother, when we grow inside the mother, from a single cell, we become a completely differentiated, a new person. So, this is how we are implanted and we are grown inside the mother. The same thing happens for cancer. Cancer actually, I will show you a movie now, cancer actually originates from a single cell. And then it divides, not only division, is completely differentiated. And it becomes a new entity within you. That is why the cancerous growth is called a somatic cell pregnancy. For a doctor, you go to the doctor. If you go to very late stage, and if you look at the cells from your mouth, you will not be able to see that these are any longer a mouth cell. If you look at the cells from your breast cancer sample, then the entire characteristics of the breast tissue has completely gone. It's a new thing now. So conceptually, this is what the state of it. Okay? Now, so this is just a theoretical definition I'm talking. The growth of abnormal cell in the body in an uncontrolled manner. Unlike, okay, this is another thing is that all tumors are not cancer. Some tumors still have a control, and that time you call it as a benign tumor. But some has no control. So when it has no control, it originated in one place and spread in another place. So that is the full-grown cancer. Historically, documentation of cancer, when first we could document. In human, the documentation of the cancer happened in 3000 BC in the Egyptian mummies. So when the mummy was, they do the X-ray, they found that several cases where the patient has, where the mummy has been, it's older people, then they could see that the bone cancer is originated there already. So bone cancer is the oldest one detected, but cancer could also be detected much, much older in the dinosaurs bone, in the fossil. So that means it is not a new disease. It is an age-old, because it has a thin connection with the irregular cell division. Okay? So I will just take you to the one important thing. One is a benign and one is a malignant. So this is what the original tumor picture I am showing you. This is what the benign tumor. And this is what you can see, there are lots of blood vessels. So this is actually the more malignant tumor. Soon you will know what it is, the malignant. So benign is often encapsulated, well differentiated, low mitotic rate, non-invasive and non-metastaticizing. And exactly opposite is the malignant tumor. You will come to know soon. So what happened? I just told you the normal cell division. Cells are dividing, but at a point cell division stops. Not only that, at a point your extra cell dies. In your body, when there is an excess cell, it will die, it will not stay. And that is how you will call this, maybe I'm blocking, maybe you will call this process is called apoptosis. In a cancerous cell, you will see there's a keep on dividing. It's not stopping. And that creates the disaster. The signal for this apoptosis, signal for stopping this, get lost because of certain mutation in the genes. What are those genes? How it happens? That's the question. That's the question we'll be hunting around. So when we talk about the cancer, there are 6 hallmarks of your body changes, of your that cells changes. And all hallmarks has one point, that the normal controls are lost. It does not obey the signal, doesn't obey the growth signal, it does not obey the apoptotic signal. That is what is happening. I'll show you a short movie on this, which will get into your mind much faster. So, these are the cells for anywhere. One of your cells gets mutated, one gene, and it has a huge rate of dividing. Look at the surrounding cells and look at these cells are dividing. Because of this mutation, these cells are rapidly dividing. And if you look at after some time, you do not need a microscope. You can see in your own naked eye a huge tumor, maybe a benign tumor is coming up. This benign tumor, growth rate is so high, it gets another mutation and it needs the food and oxygen. Blood vessel grows around it. And now it's a full-blown malignant tumor. Next step, what happens? This malignant tumor, some cells invade, you see the cancerous cells invade the blood vessels. Through the blood, this gets to some other place, far away. This is the process, and then it pops out to another place, and that gives you metastasis. Okay? So this is what the process is. Okay, you can just, for your own sake, you can see it once again. Now, because it went very fast, so mutation happened here, cells are growing very fast. Okay? Now, the surrounding cells are spread off, it's gone away. It is taking most of the nutrient. It's not sufficient. When this tumor is retaining for a long time, it's popping out. And it needs more food, more oxygen. And then the blood vessel goes and call this angiogenesis. It is supplying more, then a further mutation happens in it. So then it becomes a malignant cells. This malignant cell is much more mobile. It has the ability to penetrate the blood vessel because of new enzyme synthesis. Blood takes it from one place to another. And it's colonized in some another place. It pops out and make a metastatic tumor. Okay? So if you divide the process, it's a huge division, goes to the blood vessels, the cells, and pops out in another place. So this is the process you call abnormal cell proliferation, invasion, and then metastasis. When the abnormal proliferation is happening, a very early stage, before metastasis it can be recognized, it can be diagnosed. Then you can avoid a huge problem of cancer. But once it spreads, that's become a disaster. So now you can imagine, the drugs design where drug designing happen to stop this condition. Drug designing can happen to stop the blood vessel formation. Drug designing can happen to stop the mobility of the cells. Okay? So these are the different points where and not only that, each cases, new gene synthesis, new protein expression happens. That is what we use as a marker, detection marker. So this is what the metastasis, this is a colon to liver, original tumor image. This is original tumor picture. Original happens in the colon, but it metastasizes to liver. Breast to brain, this is a very common. Okay. These are the very common ones. A new concept. All cancer is genetic but not hereditary. Some cancer sometimes hereditary. This is what a very, very important point we have to understand. That if the cancer is to occur, there has to be a mutation in your DNA. But this need not be passing from your generation to next generation until and unless this mutation is harbored in your germ cells. So even though it is genetic cause that leads to the cancer manifestation, but it need not be hereditary in most of the cases. So how this mutation happens? This is what your DNA is. The basic reason for mutation is genomic instability. Means your DNA breakage and your DNA not repaired properly. In our everyday life, we are damaging our DNA. In every moment. Go to the sunlight, having food which you are not supposed to have, not having the food which you are supposed to take. For example, antioxidant containing food, raw vegetables, fruits, milk, proper vitamins. So this generates your, this gives a protection. And that you are damaging the DNA by the free radicals, but you are not repairing it. So that is why you induce the mutation, chromosomal rearrangement, deletion of your DNA, and your total DNA fragment gets reversed. That generates the genomic instability or mutations. So how it happens? if you look at a normal cell, each chromosome you see, you have a single color. But if you look at the cancerous cells, you have huge number of chromosomes. And look carefully, you will find out that these are mosaic color. Means chromosome has broken and rejoins. So your total gene linearity has lost. Thereby its expression has lost. So, and most of the cases it happens because of the repair system is gone. So this genomic instability, I will come to the next one, leads to the clonal selection of the cancers. So this is the normal cell. We have got a mutation. Now this cell has a much more potential to divide. You just saw the picture. In the process of division, this will be favored. And these cells will go in such a way that it will make a much more efficient to dividing a cell mass, a tumor. So, now there are 7 protein controlling genes. These genes are basically within our cell. It functions within the cell, perfect normal function. But these genes harbor the mutation because of the genomic instability. That leads to the cancerous manifestation. We call them oncogenes. So there are 2 types of genes which are normally present in our body. It doesn't come from outside. Either it loses the function or it gains more function because of the mutations. This is a very important concept of the genes and cancer. So the proto-oncogenes, which are normally present in our body, doing different kinds of function for a normal human being. These proto-oncogenes can be mutated and become oncogenes. Tumor suppressor genes, which are doing a protection function in our body. It gets mutation, it loses the function, causes the cancer. So this the opposite function of 2 sets of genes finally determine the fates of the cancer. So the oncogenes is called the gain function and tumor suppressor is the loss function. So one case it is a more function happens. Sometimes gene gets multiplied. You have more product than it is necessary, and sometimes the protection function is gone. And so the DNA cannot be repaired anymore. So there are different classification of oncogene function can be done. As I said, they are all different functions. They do different kinds of things in the cellular system: growth factor receptor, signal transduction, transcription factors. But these could be proto-oncogenes can convert into oncogene and then it do more function. That's why it supports the huge number of cell divisions. And how it can happen? It can amplify, it can go to a new place, become more active, it can have a point mutation, thereby it become completely constitutively active. But sometimes, some cases, there are some viral infection can convert this proto-oncogene to oncogene. And that leads to the cancer. So that is how some cases the virus is connected to the cancer, especially the topmost killer of this country for women is cervical cancer, which is related to the HPV. And the similar cases we find for the oral cancer. But oral cancer in South India and most of the part of the India are not due to viral. Okay? So, just now I give an example. Suppose this gene was in one place of a chromosome and due to some reason, there are different reasons, I have no time to discuss, it can come to a new place and become highly active and become multiple. Okay? So, there could be some gene because of the overexpression and then that leads to completely the cellular architecture. And this amplification leads to a new cancerous cells which has a much more number of receptors. And it looks much different than these. That is why it's much more active in the dividing and gives a much effect. So the oncogene activation can happen again from one place to another, it translocate, and in a new environment is totally different and become very active. This is the original figure what I am showing you. This is the case of the Chernobyl's, the atomic disaster. And if you see, this is the breast cancer patient chromosomes. You see, this is the normal cells, which has every chromosome has a single color. Now we look at the cancer patient, which has a yellow, red, white and blue. That means this chromosome is completely broken and rejoined. You can easily imagine, so the genes has changed its position. Proto-oncogene converted to oncogene is an active cancer-causing genes. That is inducing the tumor growth, as you have seen just now. Sometimes it happens out the oncogenes get mutated, and then it's a constitutively active. For one of the examples, the RAS genes. As I said, there are several viruses, RNA and DNA virus, which can induce the activation of proto-oncogenes. And this one is quite well known for the Rouse sarcoma virus, Penton Rouse. And this is also a fantastic work by Michael Bishop and Balmos, which actually their discovery showed how the oncogene basically get activated with the viral infections. So the next one, I was trying to tell you the tumor suppressor. The first tumor suppressor was discovered, the RB protein, retinoblastoma, and the next one is the P53. RB was discovered by Bob Weinberg, and P53 was Armin Levine. I happen to have interaction with both of them, but it has not got the Nobel Prize yet. Okay, Armin Levine, the P53 discoverer, had come to Bangalore, so I asked him that what is the Nobel Prize? So he says people have to show still that P53 is important. But P53 is the important drug target now. Okay? RB is a protein which is mainly caused for the eye cancer. Okay? And it's I'll give you a story. So I was sitting in Gallinavalli, Ganganagar, in one of the eye specialist chamber. So this doctor knows me very well. And she thinks I'm a very big guy, my photo comes in newspaper, so I should be seen first. So I told Madam, no, you see that small boy is crying. So I was sitting there, and she was looking to this boy's eyes. And so it was reflecting like a cat eye. Okay. So I asked her, Madam, is it retinoblastoma? So she said, yes. How do you know? I said, no, you put the light and it was reflecting. So it looks like there is a allele mutation. So she was very impressed on that. And then she was asking how his mother. Mother is completely normal. Then she was taking a history that anybody in your family is there or not. She said, no. So far I know there is no one. But RB, so you know we have two chromosomes. This is a fantastic gene concept. We have two chromosomes, and in one chromosome that comes from father, one comes from mother. So each chromosome we have two genes, same genes. And due to the epigenetic reason, one gene expresses, the other one does not express. So RB gene we all have. We all have RB genes. That retinoblastoma is gene, that RB genes is very important for our daily DNA damage repair. But if a mutation happens, this gene becomes suppressed. But if you have one suppressed, one fine, you don't get the disease. But if you have both mutation and suppressed, then the eye cancer happens. Apparently this mother has one mutation. She is the carrier. You will be surprised, and I got the surprise still, when doctor was asking, when you were pregnant, I told. She is not able to recall. Then she said, Madam, Jara, I had a fever. How much? She said, I had 4 or 5 fever. So what is the medicine? it seems the doctor tried different doses of different kinds of antibiotic. That leads to the another mutation. And it was a great learning there. So this is what it's a, so if it is a carrier mother, that mother got to be much more careful. So this is why the new concept of gene cheap concept is coming. I'm going to be mother. So should I not have 50 gene scan of mine? I'll give the blood, my 50 important genes will be scanned, and that will determine what I should do, what I should not do. Okay, I mean there are different diseases. For diabetes, it is coming up, risk factor. For the breast cancer, it is coming up. So the gene concept, the gene expression concept become more and more important. Well, so this is David Len, the other B53 discovered. So basically, the function of tumor suppressor in a normal cell helps with the DNA repair, but in the cancerous cells, it loses the function. Okay? I will not go through this. P53 in cancer is very important. Basically, P53 or most of the tumor suppressor are transcription factors. So, I'm tempted to show you one movie how the transcription happens. RNA synthesis happens from DNA. This is one of my favorite one. So you see, this is the P53 protein. 4 subunits together makes a tetramer. This is your gene. So this is the promoter region where the gene, this is the transcription factor, P53 comes and binds. And this is the RNA polymerase, your enzyme comes and binds here. And then the polymerase moves and your RNA gets synthesized. This is the normal function. Now, when you will find out 16% cancer, this protein P53, which you just showed, this protein is a mutated protein. It no longer can bind to the DNA. It no longer can control the transcription. But it cannot control the transcription which it's supposed to control. It controls something else. And that leads to the cancer. And that is why P53 mutation is an important drug target. You will be surprised to know the mutation of P53 for a South Indian and for a Northeast Indian are not going to be the same. It is different. The profile is different. And why is the big gap? 1.2 billion Indian. Okay? 26 ethnic group. P53 mutation are not going to be the same. And that we have to understand. There lies a big gap of science. Okay? And even the treatment also will differ. From Manipur Hospital, one radiation oncotherapist, he was talking to me and he said, Doc, can you do one thing? I'll give my patient sample 1 drop of blood. You have to tell me whether this guy has a P53 mutation or not. If so, what mutation? I said, why do you need this? He said, for a patient, for certain P53 mutation, if I give the radiation therapy, I will damage the DNA. And certain mutation is so dangerous that this patient will go worse than better. Because the DNA repair system is totally lost here. So again, I'm trying to bring up in front of you that how important is this basic science to know to go for the therapeutic point of view. Okay? Another important story is most of you know is the breast cancer and the BRCA gene. Okay? So you know this famous movie actress. She had a familial breast cancer mutation. Yes. Very well. He's so very important. And this BRCA gene mutation is actually a signature for these, and that leads to the, but BRCA mutation, BRCA1 and BRCA2 mutation, how do you correlate this? Suppose you have not checked your gene. I tell you the thumb rule. If you have seen in a family, in less than 45 years old, grandmother, mother and daughter, they have an incident of breast cancer. Even if you have seen mother and daughter as an incident of breast cancer, immediately this BRCA mutation will be checked. What is the use of it? You may say, anyway I am going to get it. No, that's not true. If anybody has a propensity of that, can have a better lifestyle, at least you can delay your breast cancer by the age of 60. Okay? So this is an important mutation, BRCA1, BRCA2, and this mutation is basically altered the gene expression function and epigenetics. So how carcinogenous genes cause the cancer? Basically it induced DNA damage and cell signaling path. And that is why it happens. A very easy thing I can tell you, the carcinogens of any form induce the DNA damage, induce the mutation, alter the cell cycle, that is the cell growth, and that leads to cancer. Anything can damage your DNA, as you see here, from your environment, from your habit of smoking, from your habit of tobacco, anything. So what are the major causes of cancer form? Tobacco, inadequate intake of fruits and vegetables, which gives you the scavenging of free radicals, excess alcohol intake, obesity, which changes your metabolic disorders, metabolic path, and a less cause also could be that too much exposure to sun. Less common cause of cancer. That could be genetics, pollution, radiation, occupational hazard. There are causes, but that is not a huge cause. Okay. These are the different types of cancer can happen in different places. For us, for an Indian, it is very important that the oral cancer and the cervical cancer, but very upcoming in India is the breast cancer. Very upcoming in India is the prostate cancer. The data for Bangalore SCG, Bangalore Manipur Hospital, Ramaya, suggests that there is a 16% increase in last 5 years in prostate cancer, even in the younger age. There is a huge increase in the breast cancer. Here the cases of is huge, and this is an alarming. Okay. Now causes of cancer. So exposure to high level of radiation, harmful ultraviolet rays, sometimes virus like hepatitis and HPV, and immune system disease. Okay. One of the important things we have to remember, I'll come to this point again, is the age risk. Why is that? As we grow old, the risk for cancer increases. The reason is what you call in scientific language called accumulative mutation. I told you that we keep damaging our DNA. We keep repairing it. But the repairing doesn't get complete. So every step we keep it, some defects. So as we grow, this accumulates. Your repairing ability comes down. Certain enzyme synthesis comes down. And thereby you induce. So age and cancer become very close. But you can reduce it. You cannot reduce your numerical age, but you can reduce your physiological age through the life habit. Okay, and that's the very reason like we are improper checkup and medical thing. We are increasing. So one of the important things is coming now is obesity. So diabetes and cancer is coming very close. So most of the cancer happens because of the metabolic reason, and that can be taken care through the proper metabolic checkup. Well, heredity is a not very big cause, and you cannot help. Obesity, as you see here, this is obese mice. There are particular genes you can mutate and make the mice obese. You can see this is a normal picture. So the huge amount of fat accumulations, fat metabolisms, that leads to this one. Now, so look at this. So So this we do not know, but definitely it is a cause of disease. If not, it is a cancer, something else. Mobile phone is a disease. So look at my friend is doing there. But I love to show this figure, the Sadhu Baba. He had with his cigarette. Here it is mobile. Commander में क्या है मुझे पता नहीं. Is it alcohol? Might be. Amma having such a huge load but has a mobile phone. So this electromagnetic radiation, the thing to be, we have to ask a question: is it inducing certain sort of glioma or the brain tumors, which we are facing these days in a very high rate as per the hospital? One of the important things is the smoke, but no less is the tobacco, in a different form of tobacco. In our country, tobacco and betel nut is taken in a different form in different parts of the country. Often we have started working with the Kolar Medical College, and there, the patients are coming. Am I audible? There the patients are coming most of the time, the grade 3 patients. So what happened? They only remove this tobacco from their cheek between the food. Even there is a tumor, to reduce the tumor pain, they keep putting inside the mouth. You tell them, whereas we are finding in Bangalore, the ECG and BIO sample, most of the sample we can get grade 1 and grade 2 samples. The samples which we get from Bangalore or Kolar or in the South India, most 80 to 90% we get the samples, they are tobacco tumors. I'm trying to make an oral cancer consortium throughout the country. So we connected with the 3 hubs. In south it is Kolar and Bangalore. In eastern part we are Bhubaneswar and northeast. So in the northeast, the majority of the cancer comes from betel nut. Okay? But looks like there is a common signaling pathway. I will just come to you in a moment. So this is one of the important reasons. These are the different things as you see the tobacco and the lung situation. These are the pictures which is collected from our own pictures, own tumors from the Bangalore Institute of Oncology. And this is, of course, I'm not going to take you through. I just give a glimpse of our work, and I'm grateful to my collaborator, Gopinath. So we do quite a bit of work on the oral cancer, and mainly we focus on the tongue and the buccal, the first inside the mouth. This is a discovery from our lab, which most likely is coming as a marker for oral cancer. This is the protein I started working, which controls the gene expression in human. And then we wanted to see what happened to this protein expression in increasing grade of oral cancer. We find that this protein is basically gets overexpressed as you go along the day. This protein gets modified also as you go along the day, a simple modification for acetylations. This is the grade 2 tumor, and we see that these are the modifications. This we isolate from the patient sample, RNA and protein, and we see this normal versus tumor, how this protein gets overexpressed. We see the modification of this is so high in the tumor. Then we look at different other epigenetic modifications, and we see that yes, this modification, what we are working is very high. We try to understand the mechanism of modification in a molecular manner, and we see this one of the enzymes, which is a key regulator of the modifications, are getting hyperactive through a method. This is the signaling pathway my lab discovered, and we know that this is the activity to be checked. So my lab synthesized a molecule which we call as the CTK7A. Several molecules we actually made. I'm not giving a scientific talk, so I'm not going to take you through. I'll just show you one molecule which is synthesized in the lab, and we show that this molecule, when you use a tumor in the mice, this is the nude mice, immune compromised mice, we take the oral cancer cell line, make the tumor, put that compound which target that enzyme which you find in the patient sample, go high, tumor growth has really come down. So lab is working toward this with the multiple molecules. So now not only we have found the target, we have also found the molecule which could be working in that direction, and we are hoping for the best for this. This is to show that yes, inside the tumor, when you keep the molecules, it has really, the enzyme activity has come down as the tumor growth has come down. Some of you might be knowing this journal, that this has come, this also covered in several people in media and all. So this is a kind of chemotherapy, you find out a new drug in chemotherapy and all. But today's concept of cancer therapy, we are coming up with a more targeted therapy. So this is the normal cell with a very few receptors. Cancerous cell has a much more number of receptors. So concept is, can we target this more so that the effect of chemotherapy comes down? How? So if you look at, this is the normal cells, this is the cancerous cells. You give a normal chemotherapy, all the cells will die. That is what you get a terrible effect of chemotherapy. If we can create a nanoparticle which will target those receptors I showed, kill only the cancerous cells, and the normal cell will be fine, that you can have a targeted therapy. This is not very far. There is a mice experiment, I will show you, that in mice we have already achieved, people have already achieved that. An important drug called Taxol, which targets the cell division tubule. So, you see this is the untreated tumor. You put the Taxol, tumor has come down, but not, it's gone completely. So you target the Taxol, you do not target the Taxol, just mix with the nanoparticle, and no, don't target the nanoparticle. This is the tumor. But if you create a nanoparticle, put the drug inside, that is the Taxol, put an arm which can bind to the cells and release the drug. You see that with the same dose of drug, the tumor is almost gone. So this is called nanoparticle driven targeted therapy. Okay? So this is what the future of the cancer therapy today, and people are thinking that how to bring it to the, few of them are in three. I will end with a problem. Problem is the concept of stem cell. At the beginning, I told you that basically cancer cell is a somatic cell pregnancy, as we all have started our life from a stem cell made by our mother and father. So cancer also actually start from a stem cell. We call them cancer stem cells. And that's the major problem of cancer therapy. How? So when I showed you a tumor, in the tumor, there are the yellows are the normal cells, but these are the cancer stem cells. You give a chemotherapy, you easily will kill these yellow cells, but you cannot kill these guys. They are very smart. They are very smart. Most of them retain the ability to come back. That is why you see there is the tumor comes back. So if we have to treat the cancer, next generation treatment, we have to target them, we have to understand them. That is why understanding of the cancer stem cell biology is very, very important. This cell's behavior is different, this cell's receptor is different, this cell's response to chemical are different. And why is the heterogeneity? Cancer stem cell for glioma from your brain is different from your oral cancer stem cells. From your breast cancer stem cells, they are not same. Because they are coming from a different tissue organism. So here lies the complexity. Here lies the science to be done before. And it becomes more as it becomes the age-dependent cancer. I told you, as we grow old, our propensity of the cancer increases. But the country which is alarming for the country is India. We are the youngest nation. This Bangalore Central School, guys, this is the auto symbol of Bangalore. Think about 25 years down the line. This young face, which is the majority of our population, a total shift of paradigm. Okay? So now, it's time for India to think, rather than any other country, to understand cancer biology more, genes and cancer more, new generation therapeutics. We cannot depend on others. We need our own research. We need our own understanding. My population is different. My epigenetics is different. I'm not American. Okay? I'm a brown Indian. Proud brown Indian. So this is a very alarming. So now the theory is coming. See, I told you present, I told you future, I want to tell you even more future. Is that, can we live with cancer? The question is, like diabetes, do you think about curing diabetes? We know. So can we live with cancer? Means cancer will be there. We will basically stop the spreading of cancer. I don't care if I have a small tumor somewhere, it doesn't disturb me. So the new concept, new therapeutic concept, new management concept has to come, that I have one operation and I stop the spread. Biotherapy by means of life. So that is the future change of strategy in the war on cancer. In summary, today I told you the gene function and cancer, how it happens. Some of the cases I talked about the new generation therapeutics, and maybe we do not need to cure cancer, but live with it like diabetes and other diseases. Thank you very much.